Good evening, everyone. Welcome to Tiny Genes, Big Effects, Hope for Rare Disease, our first event of the 2026 event series. I'm Dr. Sarah Sharman, HudsonAlpha's Research Communication Manager, and one of the voices of our science podcast, Tiny Expeditions. Tonight, we'll explore how tiny changes in our DNA can lead to rare and often devastating diseases, and how progress arises when two forces work together: basic scientific discovery and powerful technologies that let us put that knowledge into practice. Before we dive in, we would like to thank the generous sponsors who helped make this evening possible: former Mayor of Huntsville, Loretta Spencer, and Acadia Pharmaceuticals. Your support allows us to provide free events like this to engage and inspire the public, so thank you. Our speakers tonight, Dr. Greg Cooper, Dr. Susan Hyatt, and genetic counselor Whitley Kelly, study rare genetic conditions that were once considered unsolvable. Before we hear from them, though, let's take one moment to see just how far we've come in understanding disease and how progress has always depended on the partnership between discovery and technology. Let's imagine three people centuries apart who are all presenting with the same symptoms: insatiable thirst, weakness, and excessive urination. In 100 AD, Gaius, a wealthy merchant, shares his symptoms with his physician, who orders a urine test. Observing its sweet smell, he sets it outside and notices ants swarming to it. This so-called ant test leads to a diagnosis of diabetes. Without any understanding of glucose, insulin, or the pancreas, however, his doctor can offer little more than dietary recommendations and herbal remedies. We'll now fast forward to 1850s London, where we meet a young woman named Charlotte. She's also visiting her doctor for the same symptoms. This time, though, her urine sample is analyzed with a chemical solution. When that's heated, the liquid turns a cloudy orange color, which is evidence of high sugar. Charlotte still has limited treatment options, though, but she now benefits from a diagnosis that's grounded in reproducible scientific testing rather than sensory impressions. Finally, we're right here in 2026, and Maurice walks into his modern-day physician's office. Two simple tests, a fasting blood glucose and an A1C test, provide precise numerical results and a diagnosis. With a quick blood draw, his doctors can even detect antibodies that distinguish between type 1 and type 2 diabetes. These measurements enable a tailored treatment plan specific to Maurice, offering him a clear path forward. This historical journey shows us that breakthroughs in medicine rarely come from knowledge or technology alone. It's their combination that really changes everything. The fundamental discovery that sugar in urine was a marker of disease, eventually combined with chemical assays and later with advanced diagnostic tools to create the precision medicine that benefits us all today. Diabetes, however, is a well-studied and common disease, but rare diseases present a much different challenge. Unlike diabetes, they lack centuries of accumulated research, their symptoms can mimic other conditions, and their causes are hidden deep in our DNA. Until recently, we didn't have the tools or the scientific understanding to even begin searching at that level. And that's what really makes this era so exciting. Advances in basic science, like uncovering the roles of individual genes in health and development, are converging with revolutionary technological tools such as whole genome sequencing to give families answers where none existed before. Before Dr. Cooper kicks us off, let's hear from one such family who received a long-awaited diagnosis thanks to the power of genetic knowledge and technology.
So we're here sharing our story, especially us being here in Huntsville, so others can. And that's Tiana talking on her device. She's probably telling us that we're talking too much, and it's her turn to talk.
I would say things like, Tiana, come on, let's go, it's time to go, get in the truck. And she would walk outside and she would get into the truck and climb into her car seat and everything. And just one day I asked her to do that and she just stopped. She just would stand at the front of the truck like waiting for me to help her again. And I thought, come on, we've been working on this for a year, you got this. I told her physical therapist, she came over and she said, yeah, this is not okay, this is not normal for her to regress, especially after all this work.
and we went through the usual diagnostic evaluation, including brain imaging and genetic testing, chromosome analysis, and microarray, and that did not delineate the answer. And in her situation, Tiana was almost 2 when she first came to see us in neurology clinic, and the primary reason was concerns regarding her developmental delays.
When we got the diagnosis, and it was Rett syndrome, my first thought was like, oh crap, this must be serious.
This is a neurodegenerative disorder that we typically see that's almost exclusively seen in girls.
When you first find out about it, it's very painful. It's very painful. It changes your whole outlook on life, in that what you wished for your kids just may not happen. She's all smiles, all laughs, and I think I cry most of the time when she will fall and laugh, or she will not be able to grab something and laugh about it. She's just a happy girl. And so I don't know what I would do if she was not like that. She just carries me through the toughest times.
When I flip through my social media online and see someone new post that, hello, I'm so-and-so, here's my daughter, she's got Rett syndrome. Even today, I cry for them. And the one thing that, the internet doesn't give us is that I can't reach through the computer and hug them.
The honest to God truth is that everything's gonna be okay. Our life is not gonna be what we expected it to be, and that's okay.
Good evening, everybody. My name is Greg Cooper, and we're going to talk to you about some of the work that we do on rare neurogenetic conditions. And what you've just watched this video is a little glimpse into the Vega family and their journey. And she has something called Rett syndrome, which is a rare neurodevelopmental condition. And turns out there are lots of these, so we think of them as rare, and they are, but there are lots and lots of neurodevelopmental conditions. And in fact, there are so many that when you add up all these individually very rare ones, it's about 2% of children that are struggling with one of these neurodevelopmental conditions. And these symptoms can range over a variety of issues, ranging from troubles with how they grow, their ability to walk and talk, to seizures, to heart problems, a whole range of medical challenges, many of which are really, really substantial and cause a lot of difficulty. And unfortunately, for a lot of these families, we don't have an answer to some of those very basic questions of what is this condition? And answering that question is what we're talking about today, getting that diagnosis of saying this is Rett syndrome or this is another condition. Of course, diagnosis is not the final step. It's far from. It's not a cure, but it's a really critical first step on that journey for these families to help them deal with these really difficult medical challenges. So that's our goal. And that's what we're going to talk about today. But as Sarah mentioned, to get there, we have to talk a little bit about transitioning from simply observing symptoms on the outside to figuring out what's going on the inside. And in fact, human beings have long known that genetics is important, right? People can look at families and say, well, moms and dads look like their children and siblings look alike, and they often share health risks. But of course, so we know there's a genetic signal there, but it's hard to decode. It's kind of like watching TV on a rabbit ear antenna. It's very staticky, it's fuzzy. So the story we're hearing about today is how scientists have attempted to bring that genetic signal into tighter focus. And this really begins in the 1800s with an Austrian monk named Gregor Mendel, who was actually studying pea plants. And he observed patterns about how peas change over time, or change between generations, whether they're yellow or green or smooth or wrinkled. And he discovered what we now know as Mendel's laws, some of the very fundamental rules that genetics often follows. And he laid the foundation for modern genetics as a science. And what he coined the term was a hereditary factor, so the things that transmit from parents to offspring. And over the decades after that, scientists learned that hereditary factors, whatever they are, keep in mind he didn't know what it was at the molecular level, he just knew it conceptually. But scientists learned that whatever they are, they're carried on chromosomes. So chromosomes are structures that if we take any living thing, including ourselves, and take a little bit of cells and open up their cells, we can see chromosomes, these large structures inside our cells, and we knew that those were transmitting these hereditary factors. Eventually, they also learned that chromosomes are made up of DNA. So this is DNA you've heard of, is a chemical that actually has 4 very simple building blocks that we nickname A, C, G, and T. And the next big discovery about DNA is from James Watson and Francis Crick and others, who learned that DNA has this really beautiful structure called a double helix. And this double helix is made up of 2 strands of DNA, and we mentioned they're A, C, G, and T. That's all it's made up of, is long sequences of A's, C's, G's, and T's strung up together, and then 2 strands stick together to form this really beautiful and amazing structure. And this structure, all of a sudden, a whole bunch of things made sense. How DNA replicates, how it transmits information, and how it carries hereditary factors from one generation to the next. So this laid the foundation for human geneticists to really start to pinpoint specific conditions to specific genetic changes. And the first example was actually Down syndrome. So I'm sure everyone here is familiar with Down syndrome. This is a very common neurodevelopmental condition. And what scientists observed was, just looking at chromosomes under a simple microscope, that kids with Down syndrome had an extra chromosome. And by that, I mean human beings normally have 23 pairs of chromosomes. So they come in pairs because you have one set from mom and one set from dad. Kids with Down syndrome, however, have an extra chromosome, so they have a 3rd copy of their 21st chromosome. This was the first time that scientists could link a particular condition to a particular DNA change, in this case, a whole chromosome. But of course, they got a little bit lucky in the sense of you can see that with a simple microscope. But scientists also knew that if you were to drill down, if you had a very high resolution microscope, you should be able to read the A's, C's, G's, and T's that underlie those chromosomes. But we had no way to do that. So in the 1970s, a scientist named Frederick Sanger in the UK developed a technology to map out individual chemical units, to say that it's A followed by C, followed by T, followed by G. And this technology, in fact, is something we still use every day in the lab. But this allowed scientists to first start to say, okay, can we pinpoint diseases to single letter changes? And in the 1970s, people studying sickle cell anemia, which is a condition that affects how your blood transports oxygen, and scientists were able to, in fact, pinpoint a single letter swap that gave rise to sickle cell anemia. So this was the first time in human history that scientists could read out a sequence of DNA and diagnose a condition that resulted from a change to that sequence. So in the 1980s, people built on this and started to go after other well-known famous genetic conditions, including things like Huntington's and cystic fibrosis and a few others. And they made some progress, but it was very slow. And in fact, people started to realize this is way too slow. There are thousands of genetic conditions, and human genomes are big places. In fact, each one of our chromosomes has 3 billion A's, C's, G's, and T's, and doing this one at a time just wasn't going to get the job done. So some scientists, including folks like Dr. Rick Myers, who is in the audience here as a colleague and friend at HudsonAlpha, they had a bold idea. They said, why don't we sequence all 3 billion letters for one person's set of chromosomes? And this was, frankly, a crazy idea because people at the time had only been doing this one at a time, looking at a few hundred nucleotides. But it became a rallying flag, a goal that united a whole bunch of labs all across the world, much like saying we're going to put people on the moon became a rallying flag for technology in the 60s and engineering. And just like the moon landing, this fostered a huge amount of cooperation, innovation, engineering. And in fact, over the next 10 years, they were able to generate a single draft copy of one human genome. This achievement is comparable to the moon landing in the sense of it is really emblematic of what humans can do when they work together. And it was a huge achievement that transformed our understanding of the world and our place in that natural world. And since then, that has kicked off a massive and a much more dramatic acceleration of technological progress. Since the first human genome was assembled, we have made huge progress in the machinery, the fluidics, the robotics, all the things that we do, how we sequence DNA. And we've gone from taking over $1 billion in a decade to sequence one human genome to now we can sequence a whole genome in just a couple of days for around $1,000 using machines that we have at HudsonAlpha and elsewhere. And this is really what drives the work that we are doing at HudsonAlpha. So we have been harnessing these technologies, in some cases helping to push them forward and develop them. And we have used them in a variety of projects, so things like CSER or SouthSeq or the Alabama Genomic Health Initiative that you'll learn more about. And we've used them to sequence over 2,000 children and make lots and lots of discoveries about rare neurodevelopmental conditions. And we're going to talk a little bit more about that, and we'll bring up Susan Hyatt here to talk to you a little bit about how we take those technologies and translate them into diagnoses.
Hi, so Greg told us all about the technology changes that paved the way for research in his lab, but how does that work? How does it get applied? So how do we actually diagnose rare disease with genome sequencing? So I am a variant scientist as part of my job, and that means I get to look at the letters. I get to look at the A, C, T, and G for each patient. So to me, each of those letters is a data point, but to the families, it's someone looking and hoping for an answer. So when someone decides to enroll in a research study, they generally go to a clinic, a local clinic, and talk to Whitley or another genetic counselor. And they talk about what sort of features, what sort of traits the patient has, and whether or not they have a family history of any of those. They will then take a blood sample from the patient, and hopefully both parents, if they're available, and send that blood to HudsonAlpha. Once it gets to HudsonAlpha, we will isolate the DNA and sequence, as Greg mentioned. Once we have all of those letters, we will compare those A's, C's, T's, and G's to other people in the population, and we look for differences. And those differences we call variants. So these are a type of signal for us. And if you look at the left-hand side of the screen or the right-hand side of the screen, sometimes they're hard to see, but when you line them all up, most of the variants, most of the letters on the right and left side are all the same. But when you get to the middle, where there are a bunch of T's, right in the middle there's a C. So this is where someone has a letter that is different than the rest of the population. And so these variants, some of them are common and harmless, so they just represent variation among all of us. But others are rare variants, like variants we have never seen before, and a lot of those are what really cause rare disease. So again, to me, these are data points, but also to a patient, that change between just one letter is really the difference between wondering and knowing what exactly is wrong. So as Greg mentioned, we've sequenced about 2,000 individuals in our research studies at HudsonAlpha. And when I'm looking at a case, one of three things will usually happen. For about 20 or 25% of the cases, there will be a clear answer. I will find a variant that either someone has seen before or we've seen before, and we know that's just the answer. And that was the case for Tiana. In another 25%, we find a variant that maybe we think is interesting and might be an answer, but we're just really not sure. And so those cases sort of remain uncertain. But then we're left with 50%. So half of the cases do not get a very clear answer, even though we do see some variants in there. So these sort of represent a gray area, and that's really, though, where true discovery happens. And this is one of our specialties in the Cooper Lab. So to help explain what we would do in one of these cases, I'm going to walk you through one of our examples from the last few years. So this individual enrolled in one of our research studies, and she had developmental delay and autism and no family history of that. So we sequenced her and her whole family, for her and her parents, and we found a single nucleotide, a single letter change of a G to an A in a gene called RALA. And this variant, I will try to not get too into the weeds, but this variant was interesting for three reasons. First, it was very rare. No one had ever seen a change of a G to an A at this position before. It was also de novo, which means she did not inherit it from either of her parents, which is something we see often in developmental delay genes. And it was in a gene called RALA. So RALA is not actually associated with any disease, or was not associated with any disease, but some of its gene cousins are. So we knew that some related genes were associated with developmental delay. And so when we find a gene like this, where no one has associated it with disease, what we often do is submit this gene to GeneMatcher. And GeneMatcher is literally that. It is a website where you submit genes. Clinicians and researchers can submit a gene and look for other matches of people across the world that are also interested in that same gene. So we submitted RALA to GeneMatcher, and very quickly we had a hit in the Czech Republic. And so just in the course of a few hours, I emailed back and forth with a clinician who had submitted this in the Czech Republic. And we both found out that we had probands individuals with the exact same mutation in the exact same place who both had developmental delay. And so over the course of several more years, we actually found 10 more patients. And so we had this group of all of these individuals with variation in the gene Raleigh who all had similar developmental delay features. In order to really support this as a new disease gene, we also did a couple of experiments. So a graduate student in our lab, Matthew New, did some experiments in cells and showed that when you put these mutations into cells, they did change the function of the normal protein. And so all of that together then led us to be able to have a solid peer-reviewed publication. So we published this a little while ago, and this is now, everyone can now see that it is a disease gene. There's enough support for that. And so what that means is when other scientists like me are looking at their cases and see a variant in Raleigh, they now know that it is in fact a disease gene, so they will more likely flag that variant and return it to a family. And we do know that there are about 2 to 3 times as many patients who have been diagnosed with a Raleigh-related disorder since our publication. And just to sum up, I know this is just one example, and it's kind of going through it very quickly, but this is just one of many. So we have actually submitted almost 400 genes to GeneMatcher, and there are at least 35 other stories at Hudson Alpha just like this one.
Good evening, I'm Whitley, I'm one of the genetic counselors. So Susan just talked a lot about how important collaboration between scientists and clinicians are. The story about Raleigh, the other stories that she mentioned, those are all made possible because of that connectivity among those professionals. But I want to take this a step further and talk a little bit about how important connectivity can be for the patients themselves for 2 reasons. One, being the ability to connect with this technology and have access to it, but then also their ability to connect with each other and how life-changing that can be. So I don't know about you guys, but when I think of the word connectivity, I'm usually thinking about this in terms of an internet connection, right? So if you think about how internet went from being a hardwired connection between devices, allowing people to exchange information quite rapidly, and then now we've kind of progressed to things like Wi-Fi and satellite and all of these new technologies, that's kind of like how we are with increasing access to care for patients, right? So we start out and it's a very simple connection, but we're starting to utilize progress to make that a little bit sharper for them. However, it's easy to assume, for those of you that are familiar with Hudson Alpha, or maybe this is your first time coming to a Hudson Alpha event, we're constantly talking about these new technologies and these discoveries that we're making. And it's easy to assume, oh, well, if this technology is available, then it must be available to the folks that need it, right? It must be very accessible. But unfortunately, that's not always the case. There are several barriers to this. There's not one particular reason that we can't immediately roll this out to everyone that might be able to benefit from it. One of the things that we think about with this is maybe provider awareness, right? So a medical provider would need to be aware of these new technologies and what they can do for their patients so that they know that they can send their patient to a clinic or get that, access that technology for them. Another one is patient understanding or the public's understanding. Maybe a provider mentions it to them, maybe a referral's made, but the patient doesn't really understand how that's relevant to their situation or how it can benefit them. So here we can kind of think about maybe education as a signal booster for this, right? So we can maybe increase that familiarity, that awareness among the public, among our colleagues in medical spaces to understand how this could be beneficial to their patients. Another place to think about when we're thinking about these barriers are a little more logistical. So financial barriers, physical barriers. Unfortunately, this has been the case for all of genetic medicine's existence. We're kind of clustered in urban areas, so rural, underserved areas just physically don't have access to these resources. And of course, for many individuals, there is a financial component in terms of insurance or cost of getting to an appointment or obtaining the visit. So how do we increase our coverage to these network dead spots, right? So another thing, kind of getting into my analogy a little bit more, is something that you may all be aware of, even outside of genetics, just for medical research as a whole. We know that it's highly enriched over the years for individuals of European ancestry or individuals that have higher incomes or levels of education. It's both for medical research, but also just access to subspecialty care in general. And you can kind of liken this to a public versus private Wi-Fi network, right? So for the people that have the right password, access to care is relatively simple. But if you're on the public Wi-Fi connection, you don't have that password, your access is tenuous and prone to interruptions, right? So what can we do to kind of make this more accessible for all? So one thing that we do here at Hudson Alpha, in addition to what you've heard about already in terms of studying these emerging technologies, figuring out how to make use of them to help people, is also figure out creative ways to actually increase that access and remove some of those barriers. So I'm going to briefly talk about a couple of the studies. Greg mentioned them briefly in his presentation, but one of those is SouthSeek. So this is a study that Hudson Alpha was part of as part of a national consortium of evidence-generating genomic research. And we actually brought genome sequencing to NICUs across the Southeast with a focus on those individuals that were in rural or underserved areas. So about 70% of the babies that we sequenced as part of that study would have, based on their residency, been deemed as coming from a rural or underserved area. And as part of that, in addition to actually making sure they had access to the testing in their NICU, and in a lot of cases finding diagnoses for them, we also trained the non-genetics NICU providers. So these were the neonatologists, the nurse practitioners, the cardiologists that were caring for these babies. We trained them to become comfortable enough to talk about this testing with their patients and return those results. And that's important because even if the technology is available, maybe the finances are there, if you don't have a genetic counselor or a geneticist or someone that is confident discussing this testing with their patients, it may not be offered as often, right? If they don't feel confident going over the results with it. And so that training was critical to making sure that that access continued. Another study that we are continuing to work on is the Alabama Genomic Health Initiative. This is a collaboration with UAB that's actually funded by the state of Alabama, where not only are we exploring creative ways to have genomic population screening implemented into primary care clinics, but we also are utilizing an emerging technology called long-read genome sequencing that you will hear a little bit more about in a moment to increase access for individuals with rare disease across the state, so they can kind of have access to these latest technologies. And then finally, here in our own backyard, we have the Smith Family Clinic for Genomic Medicine. In addition to being a recruitment site for the Alabama Genomic Health Initiative, we're also a genetics clinic that provides clinical genetics care for patients of all ages. As part of my role there, I have the privilege of meeting with families and walking them through risk assessments, talking about genetic testing, and hopefully for them obtaining a genetic diagnosis and showing them what that looks like. And you can kind of liken this relationship to a local area network, a very specific connection that I have with my patients in order to exchange that information. And finally, one of the most rewarding parts of my job is being able to take this information once we have a diagnosis for these families and connect them with resources that are relevant to them. So being able to, ironically, use the internet in most cases as these ultra rare conditions, as you kind of saw from Susan's talk, we may be connecting these folks internationally with people that have the same diagnosis that they or their child have had made. And so building out that particular network is one of the most fulfilling aspects of my job. Finally, you've heard a lot from me about how I want to find creative ways to do my work without me having to do it, but I'm not trying to write myself out of a job just yet. Meeting with these families and hearing their stories is ultimately incredibly rewarding for me, and I hope that I continue to be a pretty reliable network provider for them. So now you're going to hear a little bit more about the Vegas that you heard the video from earlier from 8 years later.
So it's been about 6 years since we last talked to you guys. How's Tiana? What's daily life like these days?
So since we did our first update with Tiana, she's bigger. She's 12 now. She reaching almost 100 pounds, which has made life difficult and being able to handle her physically. Rett syndrome has unfortunately continued to attack a lot of her bodily functions, which just makes daily life hard. And as you can see, I have no hair anymore compared to the last video.
She has recent, within the last year, she has lost her ability to walk due to a femoral rotation in her legs. And unfortunately, fortunately, we were able to fix the rotation in her femurs through a surgery. However, putting her body through physical therapy has been pretty complicated. So her dealing with a massive loss in the ability to walk, along with hitting puberty and being nonverbal, it's been pretty rough for her. So that's what stage we're in right now.
So her diagnosis, did it function as a roadmap for you guys, or did it present any challenges, or did it bring blessings? How did the diagnosis help your family?
So I think it was extremely helpful. I know that in the beginning it's probably hard to determine whether you want that or not, but for us and for her, it has given us the confidence to find the specialist that she needs and the care that she needs. And it's also given us the confidence to advocate for her, now knowing what she needs. And on top of that, being able to understand her diagnosis and the fact that it is progressive, I will say that although it's still a punch in the gut, when she does hit her regressions, it is something that we can handle and manage and cope with, knowing that these things are coming. And so how can we care for her with the right specialists and things like that has been actually a blessing for sure.
One thing with having her diagnosis is that when we go search doctors, especially specialists of anything she may need, we're able to directly ask the questions. Have you treated someone with Rett syndrome? What's your experience with, complex needs, etc.? And I think that helps on both ends because, we've had doctors that say, oh, I'm sorry, I'm not comfortable with that, right? And let me recommend you to someone else. Now, that's difficult for us as a family because it means that we have to, continue searching at times. But I am glad that we're able to say, this is what she has, this is her exact needs, and have doctors be honest with us at times to say, I'm not comfortable with that, but here's someone else maybe you should talk to.
I will say that that the hardest part about a diagnosis is the question, what's next? And I think that being so quickly, I rave about the genetic counselors all the time at HudsonAlpha. They just helped us so much in getting connected with the Rett community. And we are still today in contact with them almost on a daily basis. And we get so much knowledge and so much experience and help through just talking through our community, whether it's just emotional help or just trying to find answers that are actually shouldn't be so difficult to find, but with the accumulative effort of all the families going through and doing their research, we're able to help each other out so much. And like I said, we are in contact with each other daily throughout the entire state of Alabama and around the world. And that's been a huge, huge help. And I know that there are a lot of families who get diagnoses and probably don't feel comfortable reaching out to other families. But if you are that person, I would say to step out of that comfort zone, because you are going to find so much help and so much support that way.
So you've heard a little bit about the work that has happened here and really all over the world, and it's a lot of progress and exciting progress. So from the 1960s, when we could only diagnose a single genetic condition, to today where we can diagnose thousands of them. I'm just at HudsonAlpha alone, We've sequenced 2,000 children, diagnosing over 600 with more every day. And that is progress that we are proud of, that we celebrate, and that is really exciting. But we also realize there's a lot of questions that we still can't answer. And that's really what motivates us, gets us out of bed in the morning, is we know there's a lot of families that we can't help yet, and we'd like to be able to figure out a way to do that. So I want to talk to you a little bit about what the future looks like and the work that we're doing to find those unanswered questions and answer them. So the first area is actually, as Whitley mentioned, sometimes it's not about the technology, it's about access. And so if you imagine children, they have this tendency to grow old and become adults. And if you, like me, are in your 40s, or if you're older, in your 50s or 60s, and you have a neurodevelopmental condition, when you were a child, there was no such thing as genome sequencing. The technology simply didn't exist. And now you might not be aware of it, your doctor might not be aware of it, and even if you are, many insurance providers won't cover it for you as an adult. So in fact, adults, especially older adults that have these conditions, are kind of shut out from genomic testing right now. So this is one area that we are working hard. We're starting a project to fill that gap. We think this is important in large part because they have questions that they'd like answers to, just like families of children do, and it might help their daily lives a little bit, become that first step on that journey to better health. But it's actually also important for children. And in fact, so we talked about these thousands of rare conditions that we have discovered in the last few years, and they are overwhelmingly discovered in children because that's where our research is really focused on historically. But what that means is, so for many of these conditions, we identify, so we heard about Raleigh, there was 10 kids all under the age of 18. So imagine you're the parent of a 2-year-old that gets diagnosed with Raleigh or another very rare condition, and Whitley or a doctor or somebody else talks to you and says, well, everybody in the world that's known to have that condition is a child. So you might have questions like, will they be independent in adulthood? Will they be able to have children of their own? Even something very basic like, how long might my child live? We can't give you an answer for that, not because those adults aren't out there, but because we've never tested them. So we want to reach out to adults, especially older adults that have developmental conditions, because again, to help them, but also because it helps everybody, including children, and it helps us give them better care, better answers to some of these questions. So the second area does relate to this technology. So Whitley mentioned something called long-read sequencing. And so the last 15 years have really, it's really hard to overstate how dramatic the acceleration in genome sequencing technology has been. But the workhorse has been something that we call short-read sequencing. And all that means is that when we talk about those DNA letters, we're looking at a couple hundred letters at a time. We actually take your DNA, your genome, and we chop it up into a bunch of 300 nucleotide, 300 letter pieces. And then we read all those 300 letter pieces. But what we really want to understand is your genome. So we want to know your chromosome structure. So those big things that are millions of letters long. And so what we have to do is basically take those short reads, those tiny little puzzle pieces, and reassemble the big puzzle that is your actual genome. And just like a puzzle, there are often regions of your puzzle that are all one color. And so you don't know how all the pieces fit and where to put them. So long reads are the answer to that. Long reads give us much bigger puzzle pieces. So instead of 300 bases, we might look at 25,000. So now imagine you have a puzzle that's 5,000 pieces and I chop it up into 50 instead. So much like how that 50 puzzle piece is much easier to assemble, you can do it faster, you can do it more accurately. Long reads give us that same edge when it comes to looking at genomes of patients with rare diseases. And in fact, we've recently piloted a technology of long-read sequencing technology. And what we found is that about 5 to 10% of children that we couldn't diagnose with short reads, we can now find an answer in long reads. So it has a really big effect, and we're excited about that and really driving that forward through all of our future work. And the last thing that I'll talk about is software, right? So we generate huge amounts of data. Every genome has 3 billion letters in it. We look at thousands of people. It's a huge amount of data that obviously we can't do this manually. So software and computers are fundamental to everything we do. And in fact, outside of Matt New, who did a little bit of experiments, my group is all computational. We are at cubicles, we stare at spreadsheets and computers all day. So we write and develop software and collaborate with programmers to build the tools that we need to handle the data, to process it, to essentially go through all these billions and millions of bits of data and to find the ones that really matter to a family's health. And that requires both biological knowledge, but also software development. And so we are constantly innovating in that space and taking advantage of innovations that come out of the digital world. So one recent example is working with Scott Newberry, who's the head of our software development team at HudsonAlpha. We received a grant from Amazon to move one of our core software packages up to the cloud and integrate a bunch of AI tools into that software. So not only will this make us more efficient and faster, but it hopefully, and we're already seeing some really exciting results, it'll make us smarter. It'll help us see the data in a different way and find diagnoses that we couldn't find before. So we're really excited about that. So just keep in mind, it's not just a science of how the chemistry of how we manipulate DNA, but it's also how we tell the computers to help us pull the information that really matters. So thank you all so much for being here. This is really, we're so motivated to work on solving these, answering some of these questions. Again, it's keep in mind that you've heard the Vega family, it's not the end of the journey, but it's a really critical first start. And we continue to try to find more and more of those answers. We're excited to be a part of that mission, and we thank you all for being a part of it. And to close up for the evening, I'll ask Jeff Moore to come up to the stage and have a few closing thoughts.
Thank you, Greg. Good evening, everyone. My name is Jeff Moore, and I am the Director of Planned Giving and Major Gift Officers here at HudsonAlpha. I have that privilege. As I listened tonight from Tiana's journey to discoveries coming from Greg, Susan, and Whitley, I'm reminded that every breakthrough here begins with a family, whether it's a parent that's searched a long time for answers for their children, or if it's that child that has been to more medical events than they have birthday parties. A family like the Vegas, who came to HudsonAlpha hoping that somewhere in that 3 billion DNA letters that we might find an answer. When a diagnosis finally comes, it isn't just scientific progress, it's relief, it's community, and it's hope all rolled in that individual moment. This happens because people like you believe that knowledge and compassion together can change lives. Tonight you've seen what HudsonAlpha does best, between pairing brilliant science with powerful technology to bring clarity to what once was uncertainty. However, discovery doesn't end here. For every family we've helped, there's hundreds more out there that need the help and need the support. And with your help, we become close to that every day. So whether you choose to make a gift tonight online through the QR code that you see on the screens, or if you send a check in, or possibly even you think longer term and you're looking at legacy giving opportunities, please know that generosity fuels the work we do here. Helping more children, more parents, and more adults finally find out the why. Your gift doesn't just fund research, it writes the next chapter of hope for families who have waited too long for that hope. So I want to thank you for being with us tonight, for believing in HudsonAlpha, and standing alongside the family whose lives you helped transform. Good night and have a safe drive home.